Good afternoon, everyone. So, for today, we will be discussing the gene therapy and natural environment. So, let's start. So, overview, brief explanations of gene therapy. First, undergo in gene therapy, importance of gene therapy in modern medicine, purpose of the presentation, dilemma or ethical issues of gene therapy, and examples of genetic disease. So, gene therapy is the introductions of genes into existing cells to prevent cure a wide range of disease. It is a technique for correcting defective genes responsible for disease development. The first approved gene therapy experiment occurred on September 14, 1990 in US when Ashanti De Silva was treated for ADA-SCID. So, gene therapy is a cutting-edge medical approach aimed at treating or preventing diseases by modifying or replacing faulty genes. It involves delivering genetic material into a patient's cells to correct genetic disorders, boost the immune system, or fight diseases such as cancer. So, ADA-SCID, it is a rare inherited disorder in which the immune system is damaged, causing a person to have a complete lack of B lymphocytes and T lymphocytes. It is a type of white blood cells that help the body fight infection. Types of gene therapy. Somatic cell gene therapy, it is a therapeutic genes transferred into the somatic cells. Like genes into bone marrow cells, blood cells, or skin cells, and etc. And it will not be inherited later generations. At present, all researches directed to correct genetic defects in somatic cells. So, somatic cells get gene therapy. It involves the introductions of novel genetic material into somatic cells to express therapeutic gene products. Germline gene therapy. Therapeutic genes transferred into the germ cells. And genes introduced into eggs and sperms. It is heritable and passed on to later generations. And for safety, ethical, and technical reasons, it is not being attempted at present. So, germline gene therapy, it is a DNA which inserted into the reproductive cells or eggs or sperm in the human body. Germline gene therapy will correct the genetic variants of the reproductive cells of an individual, and this would be passed down to future generations. Approaches in gene therapy. In vivo gene therapy, it is directly delivery of genes into the cells of particular tissue in the body. So it is a strategy in which genetic material, usually in the form of DNA, is applied to modify the genetic rep- repertoire of target cells for therapeutic goals. So, ex vivo gene therapy, transfer of genes to cultured cells and reinsertion. It is involves the genetic modifications of cells outside of the body to produce therapeutic factors and their subsequent transplantation back into patients. Ex vivo gene therapy isolate cells with genetic defect from a patient, grow the cells in culture, introduce the therapeutic genes, select genetically corrected cells and grow, and transplant the modified cells to the patient. Examples of ex vivo gene therapy. First, gene therapy to correct deficiency of enzyme adenosine deaminase (ADA), performed on a 4 years old Ashanti De Silva. So Ashanti De Silva, the first patient cured by gene therapy, and French Anderson, who planned and performed her treatment in 1990, was suffering from SCID, severe combined immunodeficiency, caused due to defect in gene coding for ADA. Deoxyadenosine accumulate and destroy T lymphocytes and disrupts immunity, suffer from infectious disease, and die at a young age. Next reporter, please.
Good afternoon, everyone. So, in vivo gene therapy, vivo gene therapy is a cutting-edge medical treatment that involves delivering a therapeutic gene directly into target cells in the patient's body. This technique is a major advancement in the field of gene therapy and has the potential to treat a wide range of genetic disorders. Unlike some other gene therapy approach that involve modifying cells outside of the body, in vivo gene therapy allows for a more delivery of the therapeutic gene, which can improve its effectiveness. Next. Example in vivo gene therapy. Therapy for cystic fibrosis. In the example of cystic fibrosis, the gene therapy involves replacing the defective gene that caused the disease with a healthy copy of genetic material using a delivery vehicle called a vector. In this case, a vector uses an adenovirus which is modified to contain the genetic material that encodes for the cystic fibrosis transmembrane regulator protein. Next. Vectors in gene therapy. In gene therapy, vectors are used to deliver genetical material to target cells. We have two main classes of vectors, which is first is viral vectors. These are different from virus, which adapt to carry and deliver genes into target cells. Viral vectors are the most commonly used vehicle by cells in gene therapy because of their ability to infect target cells and deliver the therapeutic genes efficiently. Second is non-viral vectors. These are synthetic, which do not driven from virus. Non-viral vectors are more stable and less toxic than viral vectors, but they have a lower gene transfer efficiently. Each vector class has its own advantage and disadvantage, and the choice of vectors type depends on the specific application and the target cells tissue involved in vivo gene therapy. Commonly used viral vectors, while ex vivo gene therapy can use either viral or non-viral vectors. Next. Viral vectors. Retrovirus vector system. Retroviruses are a type of virus that infects cells, their genetic material into the host cell's DNA. In gene therapy, retroviruses can use as viral vectors to deliver therapeutic genes to cells target, achieving a high level of genetic modification in target cells. Using retrovirus can be challenging as the size of the therapeutic gene that can be delivered in this way is limited to less than 3.4 kb. Next. Second is adenoviruses vector system. The adenovirus vector system is one of the most commonly used viral vectors in gene therapy. Adenoviruses are the group of viruses that can cause a commonly cold and another respiratory disease in humans. In gene therapy, adenoviruses are commodified to include a gene of interest that is delivered to target cells. One of the main advantages of adenoviruses vectors is their ability to infect non-dividing cells. It is important for the delivery of genes to cells in organs such as the liver and muscle, which contain a large proportion of cells. Next. 3 is adeno-associated virus vector. Adeno-associated virus are a type of non-pathogenic virus that can be deliver therapeutic genes into target cells. They have a single-stranded DNA and can integrate into chromosomes 19 in host cells, which can help to ensure that a therapeutic gene is stably expressed in the target cells. 4 is Herpes simplex virus vector. In Herpes simplex virus is another type of virus that can be caused as a vector for gene therapy. Infects in nervous cells, which makes in a useful vector for treating disease or the nervous system. Next. Non-verbal vectors is an alternative method for delivering genetic material to target cells that do not involve using viruses. While they have the advantage of being less toxic than viral vectors, they are generally less efficient and require higher doses of genetic material to achieve comfortable levels of gene expressions. Pure DNA construct. Pure DNA construct is a method involves delivering a pure DNA construct direct into the target tissue. While it's a straightforward approach, it has several limitations. The efficiency of DNA uptake by cells and the expression level of the therapeutic gene are often relatively low, which means that large of DNA have to injected rapidly to achieve the desired effect. Lastly is lipoplex are lipid-based formulation that are designed to protect the genetic material from degradation in the body to deliver it to the target cells. While there can be effective, most from the lipoplex get degraded by liposomes, which can compromise their efficiency. Next reporter is Smart.
Good day, everyone. So the third non-viral vectors is the DNA molecular conjugates. So the DNA molecular conjugates are molecules where DNA is covalently linked to another molecule such as protein, peptide, or small molecule. These conjugates are often used in biological and medical research for various purposes, including studying protein DNA interactions, drug delivery, novel biomaterials. So, conjugate one of A to other molecules can be through chemical reactions that form stable bonds between the DNA and the target molecule. So DNA commonly used synthetic conjugate is polylysine bound to specific target cell receptor. So polylysine is a synthetic molecule that can bind to specific receptor on the surface of target cells. This binding is often specific to certain cell types, allowing for targeted delivery. So therapeutic DNA is then made to combine with the conjugate to form a complex. The therapeutic DNA, which could be a gene or gene editing tool, like CRISPR, is combined with the polylysine conjugate to form a complex. So this complex protects the DNA from being broken down by enzymes and lysosomes, which are cellular organelles involved in digestion and recycling of their components. So it avoids lysosomal breakdown of DNA. By forming a complex with the polylysine conjugate, the therapeutic DNA is shielded from lysosomal enzymes. This allows the DNA to reach its targets within the cell intact, where it can then exert its therapeutic effects. So overall, this approach of using a synthetic conjugate helped to enhance the delivery of DNA to target cells, improving the effectiveness of gene therapy and other related treatments. So the last non-viral vectors is the human artificial chromosomes, or the HAC. So HAC is a synthetic chromosome that is designed and engineered in the laboratory to mimic the structure and function of natural chromosomes. Unlike natural chromosomes, which contain many genes and regulatory elements, essential cell function, HACs are usually designed to carry specific genes or genetic elements for research or therapeutic purposes. Also, HACs are used in various applications, including gene therapy, where they can be used to deliver therapeutic genes to cells without integrating into the host genome. They can also be used to study chromosome structure and function, as well as to create models of genetic diseases. HACs are typically created by combining specific DNA sequences and proteins that are required for the chromosome to replicate and segregate correctly during cell division. So HAC now can carry a large DNA with one or more therapeutic genes for regulatory elements. So by combining the large DNA fragments containing therapeutic genes with their regulatory elements, HACs offer a versatile platform for gene therapy and other genetic engineering applications. So they provide a means to deliver complex genetic payloads to cell or organisms, potentially offering a new avenues for treating genetic disorders, cancer, and other diseases. Next po. So next, these are the methods of gene therapy. First is the gene gun. Gene gun, also known as Biolistic Particle Delivery System, is a device used in genetic engineering to deliver exogenous DNA or DNA from external source into the cells of plants, animals, or microorganisms. The gene gun works by propelling microscopic particles coated with DNA at high speed into the target cells. So these are the basic components of gene gun, including the microparticles. These are the typically gold or tungsten particles that are coated with the DNA of interest. So the DNA-coated particle serves as carriers for delivering the DNA into the target cells. Second one is the propellant. The gene gun uses the propellant such as compressed helium to generate the force needed to propel the microparticles into the target cells. So the last one is the stopping plate. This is a plate placed behind the target cells to stop the microparticles after they have been propelled into the cells. So the gene gun is particularly for delivering DNA into plant cells, which have a tough cells wall that is difficult to penetrate using other methods. It can also be used to deliver DNA into animal cells and microorganisms. So the gene gun has been used in a wide range of applications, including creating genetically modified crops, developing animal models for studying human diseases, and gene therapy research. So gene guns employs a high pressure delivery system to shoot tissue with gold or tungsten particles that are coated with DNA bound. So next, the microinjection. So what is microinjection, Yada? Microinjection is technique used in molecular biology and genetics to introduce a substance such as DNA, RNA, proteins, or drugs into a single cell. The technique involves using a fine glass micropipette to deliver the substance directly into the cytoplasm or nucleus of the cell. So microinjection is a powerful technique that allows researchers to manipulate the genetic material and cellular processes of individual cells. It is commonly used in a variety of applications, including creating transgenic organisms, studying gene function, and delivering therapeutic agents for gene therapy. Next po. So next is the chemical methods. First is the using detergent mixtures. By using detergent mixtures, also known as chemical transfection, is a method used to introduce nucleic acid such as DNA or RNA into cells. These techniques involve the use of chemical compounds, often scatonic liquids or calcium phosphate, to disrupt the cell membrane temporarily, allowing the nucleic acid to enter the cell. So the detergent mixture interacts with the cell membrane, causing it to become more permeable or facilitating the uptake of the nucleic acid. This method is commonly used in molecular biology and biotechnology for research purposes, such as gene expression studies and gene editing. So using detergent mixtures, certain charged chemical compounds like calcium phosphates are mixed with functional cDNA or desired function. This method involves mixing positively charged calcium phosphate compounds with functional cDNA. The mixture is then introduced to cells where the calcium phosphate helps deliver the cDNA into the cells. Once inside, the cDNA can be expressed to perform a desired function, such as producing a specific protein. So next, the mixture is introduced near the vicinity of recipient cells. So this step refers to the introduction of the mixture containing the charged chemical compounds and functional cDNA close to the recipient cells. So the goal is to bring the mixture into close proximity to the cells so that the chemical compounds can interact with the cell membrane and facilitate the entry of cDNA into the cells. So this can be done by adding the mixture directly to the culture medium surrounding the cells, or by other methods that allow the mixture to come into contact with the cells. So the chemical disturbs the cell membrane, widens the pore size, and allows DNA to pass through the cells. So this step describes how the charged chemical compounds in the mixture interact with the cell membrane to facilitate the entry of cDNA into the cells. So next is the liposuction. Liposuction is a commonly used method for introducing nucleic acid into a wide variety of cells, including mammalian cells, plant cells, and bacterial cells. It is often used in research to study gene function, gene regulation, and to produce recombinant proteins. So lipofection, it is a technique used to inject genetic materials into a cell by means of liposomes. The statement described lipofections, a technique used to introduce genetic materials such as DNA or RNA into cells of using liposomes. Also, lipofection is a widely used method in molecular biology and biotechnology for introducing genetic materials into cells for various research and therapeutic purposes. Next, liposomes are artificially phospholipid vesicles used to deliver a variety of molecules, including DNA, into the cells. Liposomes are artificially created vesicles made of phospholipids similar to the structure of cell membranes. They are used as vehicle to deliver various molecules, including DNA, into cells. Also, liposomes are a versatile delivery vehicle that can be used to deliver a wide range of molecules into cells by making them valuable tools in biomedical research and therapy. Other types of gene therapy. First is the gene augmentation therapy. Gene augmentation therapy is a type of gene therapy that aims to treat genetic disorder by supplementing a functional copy of a gene to replace or supplement a faulty gene. So the goal is to restore the normal function of the gene and alleviate the symptoms of genetic disorder. So gene augmentation therapy, or GAT, most common form of gene therapy. So this approach known as gene replacement therapy. It is one of the most common forms of gene therapy. It involves introducing functional copy of gene into cells to replace a missing or defective gene. So foreign gene replaces missing or defective gene. In this context, a foreign gene refers to a gene that is not normally present in patient's genome. The foreign gene is introduced in the patient cells using a vector such as virus or lipid nanoparticle. So replacement of defective P53 gene by normal one in liver cancer. So in liver cancer, the P53 gene, which is a tumor suppressor gene, it's often mutated or defective. This mutation can lead to uncontrolled cell. This mutation can lead to uncontrolled cell growth and cancer development. So gene therapy for liver cancer may involve replacing the defective P53 gene with a normal functional copy of the gene. So next is the gene inhibition therapy. So gene inhibition therapy, also known as gene silencing therapy, is a type of gene therapy that aims to treat diseases by reducing or inhibiting the expression of specific genes. This approach is often used to target genes that are overactive or mutated and contribute to disease development. So there are two types of gene inhibition therapy. First one is the anti-gene and the second one is antisense. So anti-gene therapy is a type of gene therapy that aims to inhibit the expression of a specific gene by targeting its regulatory sequences. So the goal of anti-gene therapy is the blood transcription of the target gene, preventing the production of its protein product. So anti-gene therapy typically involves the use of synthetic oligonucleotides that are complementary to the regulatory sequences of target gene. These oligonucleotides bind the target gene's regulatory sequences such as promoter regions or enhancers and block the binding of transcription factors or other regulatory proteins. This interference prevents the initiation of progressions of transcription, effectively silencing the target gene. So next is the antisense. Antisense therapy is a type of treatment that uses short single-stranded nucleic acid or the antisense oligonucleotides to target and inhibit the expression of specific genes. So these oligonucleotides are designed to be complementary to the messenger RNA or the mRNA produced by the target genes. By binding the mRNA, and antisense oligonucleotides can block the translation of the mRNA into protein, so effectively silencing the target gene. So antisense therapy can be used to treat a variety of diseases, including genetic disorder, viral infections, and certain types of cancer. It is often used to target genes that are overexpressed or mutated in these diseases. Antisense oligonucleotides can be delivered to cells in various ways, such as through injection or infusion, and then can be designed to target specific tissues or organs. Next. Next is the disadvantages of gene therapy. First is the long-lasting therapy is not achieved by gene therapy. Due to rapid dividing of cell, benefits of gene therapy is short-lived. Next is the immune response to the transferred gene stimulates a potential risk to gene therapy. Third, the viruses used as vector for gene therapy. Gene transfer may cause toxicity, immune responses, and inflammatory reactions in the host. Lastly, is the disorder caused by defects in multiple genes cannot be treated effectively using gene therapy. So, despite these limitations, gene therapy has shown significant promise in treating a variety of genetic disorders and has the potential to revolutionize the treating of many diseases. Ongoing research is focused on overcoming these challenges to improve the safety and effectiveness of the gene therapy. So next po. So these are the advantages of gene therapy. First is the gene therapy has the potential to eliminate and prevent hereditary diseases such as cystic fibrosis or the ADA, SCID, or etc. Next, it is a possible cure for heart diseases, AIDS, and cancers. It gives someone born with genetic diseases a chance to live. Lastly, it can be used to eradicate diseases from the future generations. So overall, gene therapy has the potential to revolutionize the treatment of genetic diseases and provide new options for the treatment of complex diseases. Ongoing research and development are focused on improving the safety and effectiveness of gene therapy to realize its full potential in clinical practices. So, recent developments. In a new gene therapy method developed by University of Florida in January 2012, researchers found treatment for a common form of blindness, which is the X-linked retinitis pigmentosa, that strikes both youngsters and adults. So, the University of Florida developed gene therapy methods in January 2012 to treat the X-linked retinitis pigmentosa, or the XLRP. a form of inherited retinal degeneration that causes progressive vision loss. So ano daw siya, namamana siya. So the therapy involves delivering a normal copy of RPGR gene into the retina using a adeno-associated virus or the AAV vector. This approach aims to restore the function of retinal cells affected by the disease, potentially halving or reversing vision loss. So, a gene therapy now called NLX P101 dramatically reduces movement impairment in Parkinson's patients. According to the result of phase 2 studies published on March 2011 in the journal Lancet Neurology. So the NLX-P101 is a gene therapy that has shown promising results in reducing movement impairments in Parkinson's disease patients. A phase 2 study published in March 2011 in the journal Lancet Neurology reported that NLX-P101 significantly improved motor function in Parkinson's patients. The therapy involves delivering a gene called glutamatic acid decarboxylase or the GAD into the subthalamic nucleus of the brain using a viral vector. So the GAD is an enzyme involved in the production of gamma aminobutyric acid or the GABA, a neurotransmitter that helps regulate motor function by increasing GABA levels in the brain. So the NLX-P101 aims to improve motor function in Parkinson's patients. Conclusion. So theoretically, gene therapy is a permanent solution for genetic diseases, but it has several complexities. At its current stage, it's not accessible to most people due to its huge cost. A breakthrough may come anytime in a day, may come when almost every diseases will have a gene therapy. So gene therapy has the potential to revolutionize the practice of medicine. Overall, gene therapy holds the promise of being a permanent solution for genetic diseases, but it faces complexities and high costs that limits its accessibilities. Breakthroughs could make gene therapy more widely available, potentially revolutionizing medicine by offering new treatments for a range of diseases.